So, welcome everyone for this afternoon's JChem for Excel presentation. Actually, this is the second presentation here in the US. And what I would like to focus in this presentation, actually just to give you numerous examples how you could use, so how intuitively you can use JChem for Excel in the drug discovery fields. And I'm also going to talk about the recent improvements and the future development parts in plans in JChem for Excel. Actually, I'm going to have another short session after Charlie, and that one is going to focus on how is it possible to extend, interface, and enhance JChem for Excel. So the second presentation is going to be more focused for developers. So what's new actually in these new 5.3 versions? Actually, as you may have noticed, we changed the versioning of JChem for Excel. So we are now have the same version as the underlying Marvin and the JChem API. So many evaluators and customers actually requested that we should be more flexible. And that is the reason we decided to be able to add various third-party editors and also third-party cartridges. So what we have now at the moment, we also support ChemDraw, IsisDraw, and ChemAxon for editing. So in case you use these editors, you can do that with JChem for Excel. We also started the support for third-party cartridges. At the moment, we worked with ChemAxon, or the older MDL direct cartridge, and we also have a support for IDBS cartridge. Probably these cartridges will be available later on as plugins under a different licensing scheme. You will have to discuss this with the responsible salesperson. So I'm just looking at the technical perspective of these features. The third point is quite technical, but it's also a milestone in JChem for Excel development. So we also added the so-called Marvin OLA support to Marvin.net. So this means it is possible with Marvin.net to copy structures into Office documents and edit them later on. And why is it important? Because so far we needed the capability of the Java-based Marvin beans for that. And after we made it in Marvin.net, there is no longer need for any Java to use the full feature set of JChem for Excel. We also introduced the customizable UI. I'm going to talk about it a bit more detail in the development session. At the moment, it is only available with XML, so it's mainly for administrators and developers and not for end users, but later on we would like to also add visual interface for that. We also added many smaller enhancements. One of them is now it is possible to do R group decomposition on the same sheet. It was requested quite often. And we also added the numerous actually features working with OLE objects. These are quite commonly requested features because this is the basic model how you interact with other Office applications. So if you want to copy over structures into Word or PowerPoint, edit them there, copy back, and so on, you will have to do this with OLE objects. So I'm going to give you several demonstrations how we think you can use JChem for Excel to accomplish various tasks. Oops. So the first one is titled actually Compound Profiling. I'm going to open up the necessary file. So actually, I already prepared this spreadsheet. I'm not going to do this here on the fly. So what you can see here, Do not worry about how the structures are displayed. We actually have a lot of discussions about this, that we should clean the demo data we use, but at the moment it is just a structure set imported from an SD file. But in a later example, I will show you how you can actually accomplish this. So we have a set of structures, and actually in the other columns from B to H, we have various function calculations. So actually what you can see here are all dynamically calculated functions. What I put in the first row is basically a threshold of the results for these calculations. There are actually many thresholds available for predicting drug likeness and so on, the Lipinski rule of 5. Actually, I put one of them there. And what can you see under that? There is a conditional formatting. So those structures which do not match these criteria are highlighted in red. So basically, in this way, if you use this template or function set, it's very easy to oversee your compound library and actually check which structures do not match the criteria you specified. Okay. In the next example, I'm going to look at the scaffold structure for the structures, and I'm going to sort that basically on the complexity of this scaffold structure. So here I have only just a small number of structures for illustration. And in column B, I'm going to put up a function which will display the scaffold structure for me using the Bemis-Murko method. So this is A2. You have to be very precise with this double click to filling the structure. So if your hand's a bit shaking, it won't work. As you can see, it displayed the structured scaffold for me. And based on this calculated structure, you can also do further calculations. So now I'm going to calculate the heavy atom count of the scaffold structure. That's it, it's 6. And of course, you can also sort on this data. And now it will display the structures in so-called complexity order, so based on the heavy atom count of the scaffold structure. So the next example is just a very easy example, just to show you how you can visualize some things using the Excel charts. So I have a structure caffeine in A1. These are basically the same thresholds and calculations which you saw in the first example. So what is interesting here is just the radar chart from the Excel chart library. And what it displays actually that in red you can see the threshold values, and in blue you can see the actual values. So actually this structure fits into this criteria. Actually, what is not really good with the built-in Excel radar chart, that it's not possible to define the scale range of the various axes of the radar chart. So we will have a look at the new Excel version, and if it still does not support it, maybe we'll also look at including some other third-party charts as well. So I'm going to close the tab. So the next example, I'm going to just quickly show how the R group decomposition to a same workbook works. I'm going to quickly open this SD file or import it. Click on R group decomposition. Select the query structure. I'm not going to draw it now, just open it from here. As you can see, I made a little hack for those who were in the user training because it did not fit in the projector area. But anyway, I quickly fixed it. So click Run. And it will place the results on the same sheet. And for those structures which are not matching the query structure, the cells will be empty. So in my next demonstration, I'm going to show you how is it possible to arrange some data in the matrix arrangement in JChem for Excel and display the similarity between various structures. So I'm going to use only this small sample for this and select all. Click Copy. And with the special paste function, transpose, actually you can paste the structures in a horizontal direction. So from here, I'm going to select dissimilarity functions. We have various dissimilarity functions. Probably Mitsuo could explain you what are the differences between these, but I'm going to pick this one. Pick the first molecule, the second one. If you want to do these matrix calculations, you would actually have to specify the absolute location for certain cells. So I'm going to put this $sign here. It means that actually it will always take the structures from the A column, and for the next one, we have to always use the first row. So it displays 0 because actually these are the similar structures. So I'm going to drag to the right, fill down. And actually, to be able to, or with help to oversee the differences or dissimilarity in these structures, you can apply the conditional formatting you prefer, and it gives you a very nice overview about the dissimilarity values. So in my next example, we are going to look at these nasty SD files, or the nasty structures from these SD files, and make them look a bit nicer. Okay, I'm going to close them up. Actually, I'm running 5 minutes, so I will hurry up a bit. I will show you only the results. Not going to do it step by step. So in the first column, I have the original structure. And actually, on the corresponding columns in B, C, D, and A, in each column, I apply the special standardization. And as you can see, for example, the function in column C takes the result of the standardization in B. So it's basically doing a step-by-step standardization or canonicalization in the structure, and you can see the result in the column A. You can also do this in one step by specifying this action string in one value separated by 2 dots. So next, just a quick example of the reactor functionality or virtual library generation of JChem for Excel. Here you can see basically products generated based on the structures in column A and in the first row in a matrix arrangement. You can see the generic reaction in A1. So basically, these are all calculated dynamically on the fly. So if you change the structures, all the products will update. Going further, you can also do calculations based on the products. So what you can see here is basically just the same structures what you see on the first worksheet. And these values are all calculated from the resulting products from the first page. So these PSE values, and also you can apply conditional formatting. You can also do additional calculations. So for example, you want to see the pKa values for the resulting products. You can also make these calculations and display it in a visual form like this. Okay, just a short overview about what's, or demonstration what's coming in 5.4. Actually, we will include the most common substructure functionality as a function again. So what you can see here is the structure set again in column A and in the first row. And in between, you can actually see the most common substructure between them using this formula. Related to Markush, we also introduced Markush match function, which actually I took this. These are simple examples. I took it from the unit test examples of Szabolcs. So what you can see here is there's some query structures here, some targets on the A column, and in these all cells in between, actually it displays whether the query matches the target or not. Okay. If you are interested, we can also give you some additional demonstrations or help what you can accomplish with this, but I'm going to continue my presentation, just give you a quick overview what's coming. I talked about a few ones, but we will soon have Office 2010 support, so my colleague informed me that it's working quite well, so hopefully we can include it pretty soon. We would like to, I forgot to demonstrate you the structure checker functionality. In recent versions, we are going to have these as structures, but later on, we would like to also include it as a general functionality as in Marvin. So as you add structures or edit them, it will dynamically display the errors, and it will be also possible to correct them. So just a quick brainstorming or ideas what we thought it would be nice to have in JChem for Excel. One of them is actually so-called refresh as you edit functionality. So we like to add the Marvin editor, rather as a dialog, but as a task pane. So you will see basically the Marvin editor next to an Excel sheet. So for example, if you click on a cell, edit the structures, it will be dynamically updated in Excel sheet. And if you have various calculations based on that, it will be also dynamically updated based on each edit action. Based on this text pane functionality, we also have some ideas what to put there. So for example, we can have a grid to display additional related structures for the selected structures. We would like to also put the filter functionality into this task pane. So if you want to refine your filter, you don't have to go to the dialog again. And if you are interested, please come to me, and I can also talk about some additional plans we have, because I'm a bit over time. Just as a final word, I would like to introduce our small team. Actually, Victor is doing the heavy lifting in JChem for Excel. He's doing all the rendering logics, the third-party editor integrations, and all this OLE stuff. András, he recently joined ChemAxon. He's also working in other products, but now he's taking part of some restructuring of this rendering. And myself, I'm also involved in development, so I usually do the features and interfacing with the ChemAxon API. Thanks. We can also do some questions now, or if we can also do it after my next presentation. If the time allows, I will leave it up to you, John.
Okay. Are there any immediate questions? Let's give a round of applause to Tamas.
Thanks.